Here we are. There it is. And then I'm going to share my screen and we'll do this thing. Slideshow, start. So welcome everybody. This is the Lower Nehalem Watershed Council Speaker Series. I'm Zach Mallon, the Council Coordinator. And you aren't here to see me talk tonight, but since I've got you here, I'm going to talk a little bit about the Watershed Council before I turn it over to Dr. Miller. So, the Lower Nehalem Watershed Council is dedicated to the protection, preservation, and enhancement of the Nehalem watershed through leadership, cooperation, and education. We accomplish this mission in this geography. You can see Highway 26 here and coming along. So we're just south of Highway 26 for most of the area. It's all of the main stem Nehalem up to the confluence with Humbug Creek, and then all of the North Fork Nehalem as well is our coverage area. We work with landowners, private companies, all stakeholders that are willing to work with us to restore and enhance habitat for fish and wildlife. We do this through research, fish passage projects, riparian plantings, in-stream enhancements, and wetland restoration. And we also do outreach and education programming like the talk here tonight. Tonight, I'm just gonna highlight a project that we're working on right now, which is our Coal Creek Riparian and In-Stream Enhancement Project. What you're seeing in the background here, the slide or on the slide, is a culvert that washed out at the start of 2021. We had luckily already slated it for replacement because it was undersized for the size stream that was going through, and serving as a velocity barrier to salmonid migrations, both for juveniles and for adults. So thankfully, when it washed out, we'd already gotten together the funding to be able to replace it. And based on the local area and the proximity of several trees, we realized that the best solution was going to be to put a bridge in instead of actually putting in a new culvert. So you can see here on the side, this form that's being used to pour the bridge. And this form is actually adjustable for different size bridges, which is a pretty neat feature of that form. And here's the finished bridge right there. So pretty exciting that we got that part done. Another big component of this project has been the riparian plantings. And these are two views of the same location, only from different sides. And you can see that the riparian area on the left-hand side is very full of a lot of blackberry, and there's a big steep slope and an undercut that's happening. So what we did is we planted the top of the slope in the top of the field here for about 15 to 20 feet in this location. And we also did a bunch of staking along the slope itself to help establish vegetation there, help build up the root mass to hold back some of that erosion, as well as to eventually shade out and outcompete some of this blackberry that we'll be maintaining for the near future. As always, if you have a project that you're interested in working with us on, feel free to let me know. Most of you found this probably through either the Facebook or our email list, so you have good ways to get a hold of me. So what I'm gonna do then, oh, I forgot to mention, here's the area. It's about a mile, about a half mile of planting that was done. But without further ado, I'm gonna pass it off to Dr. Miller here. Dr. Miller comes to us from Oregon State University and is a professor of fisheries, wildlife, and conservation science, and also leads the Marine and Anadromous Fishes Ecology Lab down in Newport. And so, Dr. Miller, if you want to take it away. Have at.
Sure. Let's see if we can do the right one here. Are you seeing the notes page or a slideshow? Let's see. Let's make sure this works.
I'm not seeing any notes, but I do see your sidebar of all the slides.
Now, which do you see? Are you seeing the actual screen or the notes?
I'm seeing notes too.
Shoot. Okay. Always a trick with this one. Let's try to share. Guess we should have tried this instead of chatting.
That'll happen. It's not the first time and it won't be the last.
That then.
It's a good looking columbine.
How's that? Okay, excellent. Great. Yeah. Excellent. Well, good evening. I see it's a pretty small crowd, so I'm happy to make this more of a conversation. It's always nice, especially when you're just staring at your screen. I was chatting with Zach. I used to work for the Tillamook Bay National Estuary Project many years ago, so I'm familiar with Nehalem. And I actually also coordinated a stream restoration project on the Bear River on the Willapa Bay, doing alder conversions and inwood placement. And a big component of the research program I have does focus on salmon and lamprey. But actually, I'm also going to talk to you today about heat waves and Gulf of Alaska Pacific Cod. I hope you find that interesting, but I also think it's relevant in the sense that, well, just marine heat waves are going to be an issue for us moving forward, and they have been in the past. And I will talk about salmon a little bit at the end, but all the mechanisms that are affecting the cod and the way they might adapt are and that have analogs with salmon and other freshwater species too. So with that, please do feel free to raise your hand or unmute yourself and ask a question as we go. That would be great. So I'm going to start with collaborators and funders because this is always a team effort. Several people, 2 postdocs and graduate students and colleagues at Oregon State University and colleagues at NOAA, the Northwest Fisheries Science Center, and the Alaska Fisheries Science Center. I work with a lot in collating and collecting samples and doing a lot of the different analyses. So I'm going to talk a little bit about why marine heat waves and what are they, how are we defining them? And then, as I mentioned, give you a sort of a run through of what happened in Gulf of Alaska Pacific Cod in response to what's often called the blob 1.0 and the blob 2.0. And then finish up with a little bit of information on the California current and Columbia River Spring Chinook salmon. So some of these may, if any of this is completely redundant for you, I apologize. Just want to get a couple terms out there so we're all on the same page. A lot of the climatology works with anomalies, which are really the difference between the current period and some reference period. It's usually standardized to some time. And so this graph is a sea surface temperature anomaly. And it's the, for example, this is early December 2020. It was a positive anomaly of about a degree, meaning it was a degree warmer than that 1998 to 2020 baseline, a 30-year baseline. And then going down, the blue lines are negative anomalies, so it's colder than normal. And then cohorts are, in this case, groups of fish that share one or more characteristics, such as hatch date or age. And recruitment can mean many different things depending on who you ask. It's a measure of how well that cohort succeeded. Usually in this case, it'll be juvenile settlers on Kodiak Island nursery areas, or the number of fish that reach a reproductive age or enter a fishery. And so starting in 2014, late 2014 into 2015, the Pacific Ocean warmed to a level never recorded. So this graphic is the monthly average sea surface temperature in May of 2015. And you can see that these areas, the darkest sort of maroon colors, is 3 degrees Celsius, over 5 degrees Fahrenheit warmer than the long-term average. And this led to a cascade of issues throughout the food web that we're still understanding and preparing for today. So I want to give you a couple different ways to see it. This was called The Blob by Nick Bond, Washington State climatologist. Coined this term as he was one of the first to really notice what was going on here in the, can you see this cursor?
Yep.
Excellent. Let's get the, let's try this one. So he noticed that pool of warming water in late 2013, which then just got deeper, larger, and warmer. And it was largely due to what he was also termed as a ridiculously resilient ridge of high pressure. So a really strong high pressure system set up over the North Pacific. So storms were not happening. And so winter mixing did not occur. And then more summer warming happened. And then it evolved and took actually 3 different forms that had different forcings. It became something called the arc pattern here in July of 2015, and then morphed into what they call the Baja pattern in late 2015 before it dissipated in 2016. And again, those anomalies were, over 3 degrees above normal. And so this graphic shows, the top plot shows that ridiculously resilient ridge of high pressure. The solid lines are contours of sea level pressure. So those are positive anomalies. And the dashed lines are negative sea pressure anomalies. And that blue star is where this temperature profile comes from. And so this ridge set up and prevented storms from reaching the water and mixing. And this blob, as they called it, you can see here, starting about 2014, those temperatures started climbing. And then by October, November, December in 2014, some odd things happened with then a strong low pressure system sort of taking over the area and helping create this arc pattern. And that's when the warming really came into our waters. And if you were around the coast in 2014, 15, 16, you probably experienced some of that happening off the coast of Oregon and Washington. And then this Baja warming pattern was the shift in late 2015 before things started cooling down again in 2016. And so what happened was a tremendous mix-up of the entire food web. And so this is a summary from Leticia Caval, who collated observations all the way up through the Gulf of Alaska. There were mass strandings of seabirds, such as brown boobies. There were shifts in distribution. So what you saw there in the red boxes are all of those shifted further north than they had been reported before, and some 1,000, 3,000, 3,600 kilometers further north than we had ever been recorded. Scalloped hammerheads, thresher sharks, wahoo, yellowtail, green sea turtles. And we also saw then a repeating of unusual records from other, say, El Nino events where the ocean got quite warm and we might have seen Humboldt squid further north. So that whole suite down here at the bottom of the graph were again seen moving farther up to those northern extents. So pretty dramatic reorganization of who was around in whose waters. And there were also other effects. There were large harmful algal blooms. The largest ever recorded harmful algal bloom, which is due to a diatom pseudonychia producing domoic acid, has now been linked to that warming event and the weather patterns that coincided to create this massive bloom that, as again, if most of you were around then, that closed the razor clam fisheries, delayed the Dungeness crab harvest. The little orange figures are dolphins and seals and whales that had detectable domoic acid, and then the red ones were actually poisonings and deaths from that. And one thing that's really relevant to salmon, as well as pretty much everything in the mid and upper trophic levels, was the copepod response. So here off our coast, traditionally upwelling summertime, we get a suite of large-bodied, fat, lipid-rich copepods all through our coastal waters, fewer larger species. And the more we have, the more biomass of northern copepods, usually the better the salmon survival, the better almost everybody survives. Then in the winter and often in what we call poor ocean conditions or warmer times, we get these communities of copepods that are smaller bodied, low fat, and many more species, greater species richness. And so this graph, each of these gray dots is one data point. And so down here in Newport, there's the Newport hydrographic line, and it goes from very close to shore out to 25 miles offshore, regular stations that an attempt to sample it monthly by NOAA and OSU colleagues, and they usually get somewhere around there. So there's this long-term time series looking at the community and how the community shifts with the water, and then how all the upper trophic levels respond to that shift in the food web. And so forage fishes like juvenile anchovies and sardines eat those zooplankton, and the yearling Chinook and the coho that enter the ocean eat those forage fishes. So there's a direct, or I guess one step removed, indirect link to these copepods. And a lot of salmon survival studies, we've often seen the survival and growth of Chinook and coho are related to these communities. And so you can see here in 2015, when the heat wave really approached our waters, this is the biomass anomaly. So there were way fewer, lower biomass of those northern copepods, those fat, rich copepods. And there was many more of those small southern copepods. So that was a system that actually is mirrored with a different suite of species, but the same cool, large, fat copepods, small, warm, skinny copepods is also seen in the Gulf of Alaska. So it's a mechanism that we see along the West Coast. All right, so to recap, what we talked about is we kind of had our what we think of as our normal upwelling summers, the wind's blowing your head off when you go out to the beach, and plenty of krill. Then 2014, there was little to no wind. The ocean was warming. The blob developed in the winter. We also, in our waters, had low snowpack. 2015, the blob really grows, expands, and becomes that arc pattern and comes into our West Coast. And also then an El Nino developed in 2016. So there's a double whammy there in 2015 and 16. And then you saw pyrosomes, maybe some of you saw them on the beach. There were massive numbers of these tunicates around, sort of taking the place of a lot of things that salmon and other animals normally eat. And there was also that year where the upwelling band was so narrow, everyone was there. The whales were there, the salmon were there, the crab pots were there, the crabbers were there. There was a lot of conflict between the whales and the crab pots and entanglements because the band of cold water was so narrow and close to shore, which is in many ways, as people have said, is this kind of warming gave us a glimpse into the future. Those are anomalies that were predicted to occur more like 2050. And so we had 2 or 3 years at a very acute level to see how the system responded and get an idea of some of the conflicts and challenges that will face us. But just as it started to cool down, Blob 2.0 came in September of 2019, or late summer. And this was different in the sense that this blob set up in the summer, not the winter. So it was sort of the opposite. There was actually less wind. Same idea that there's less wind, but it was during summer. So the surface layer, the stratified layer warmed up really fast, hot, quick, but fortunately it did dissipate pretty quickly and didn't cause another 2 or 3 year cycle. And so a lot was the same time that heat wave was happening in the Gulf of Alaska and the West Coast, major heat wave off the Indian Ocean and in Australia. And it led to a global effort to define heat waves more like we define droughts or heat waves on land, have a more common language to better understand the commonalities, the global patterns, how much of this is due to climate change. And so currently a pretty accepted definition is a heat wave is a heat wave event when there's more than 5 days of temperatures over the 90th percentile. In this case, the 30-year baseline, 1982 to 2012. So the anomalies would be in reference to that time period. And this graph gives you an idea. It's not the best, but the green line is the threshold above which it would be a heat wave. And the black squiggly line is the actual temperature. And the blue is sort of the long-term climatology. And this is from a temperature gauge just by Kodiak Island, Alaska, where I'll talk about the Pacific Cod. And you can see here in the lighter orange and then the darker red, those are the periods, the duration of the marine heat wave events, starting there in late 2014, becoming quite dramatic in 2015, and then resurging again in 2019 and a little tiny bit in 2020. And sort of the last sort of background heat wave piece is, so what's happening around the globe, this graph on your left, A, C, and E. Each of those blue lines is a documented heat wave globally, with the red lines being the 7 most largest. And they're quantifying it by the duration on the top, so the number of days it lasted, the peak temperature anomaly in the middle here, and the cumulative intensity, the cumulative degree days. And so you can see, obviously, there's more blue as we get into the present time, and they're more severe. So they are definitely increasing in frequency, intensity, and duration. And several studies have linked some proportion of that being, a result of climate change. There's 2 different future projections based on global climate models. RCP 8.5 is the less optimistic one, and that's what that picture is. And that shows this sort of basically pretty much the entire Pacific having a 3 to a 4 degree Celsius anomaly. The somewhat more optimistic projection of the 4.5 has future temperatures a little warmer and more focused. The warming is more focused actually in the northern waters. And just if you pay attention to those things and you follow those models, this just gives an idea of the 2 I referred to was this RCP 8.5 and then the 4.5. And business as usual projections is this black bar, and business as intended. So you can see that the 8 is a little pessimistic, but it's what many are sticking with because there's reasons to be pessimistic, I think. So how does this ocean warming affect fish? And when we think of who recruits to a population, who succeeds, survives to whether they make it to the dinner plate or to reproduce on their own, there's multiple hypotheses that really dovetail together. And they're the critical period hypothesis, which is the idea that most, well, almost all, unless they're direct developers like surf perch, where they pop out, fully formed little juveniles, most have a yolk sac. And so then they have to find food before, like within 2 to 7 days, depending on the species and the water temperatures. It's called the point of no return. If that larva doesn't find food to eat, it will just die. So it was termed the critical period, which is expanded to include different points in the early life history. And so when that yolk sac is being absorbed, there has to be food around. And that led to the mismatch hypothesis, the idea that if phenological shifts or like the timing of production or hatching shift and they're not overlapped anymore and there's nothing to eat or that larva can't survive, then you'll have poorer recruitment when you have a mismatch and better recruitment when you have a match between the predator and the prey. And that relates to the stage duration or this growth mortality hypothesis ideas that when you're little and you don't have good capacity to swim, you're easier to eat, more things can eat you because you're smaller. So the faster you go through your stages of development, the less you're going to die. Your mortality rate's going to decrease. So this idea that the faster you grow, get to the next stage, you'll survive better. But as you can imagine, when it gets too warm, there can be bioenergetic stress, which I'll talk a little bit more about, which can then influence your ability to find food, avoid predators, and survive. And then at all of those events, at all stages, even at the adult stage, right, there can be selective mortality, where either slow growing or smaller fish are selectively removed. They're easier to catch and eat. So then you have fewer individuals, but they're often in better shape. So all of those happen. They happen in salmon, in Pacific Cod. And just a little bit of history, because I think it's pretty interesting. The idea of this critical period is over 100 years old, 120 years old, actually. Johan Hjort is a famous fishery scientist, and also the first to have noticed, to have put things together. We didn't really understand then why fish populations went up and down. We still have trouble predicting it, but we know a little bit more why. And he was the first to age fish. So in Europe, spring herring populations just crashed. and the government paid him to go figure out why. And so he was the first to combine the ability that had just been determined by another scientist named Henke, that you could age the scale, the fish scale, and figure out how old a fish was. So he went all through the region collecting spring herring. And this old graph from his work has 3-year-old to 16-year-old fish in 1908 to 1913. And you see this huge spike in the abundance of 4-year-old fish in 1908. And he followed that year class through time, and he realized, wait a second, this means just 1 year made up the entire fishery. And once that year was fished out, so to speak, because that was most of the biomass, there was no herring left. So that led him to this critical period hypothesis that is still active and supported in many places. But as you might imagine, all of those different mechanisms can be happening to different extents. And so that's why it always ends up being a challenge to keep understanding what's happening for a particular species in a particular region at a particular time. And right, so that's really how that all those fit together, is that yolk sac stage needs to find food before the yolk sac's gone. And Pacific salmon, they obviously solve that problem by staying in the red, right? So they don't expose themselves to that getting lost, drifting, or getting eaten unless the nest is dug up until they swim up. And then we call it a preflection stage, because here the caudal fin is not formed yet. It's just basically like a little extension of the notochord. So they don't have the best swimming capacity, but they need to avoid predators and find food. And then that flexion stage, they have more ability, and finally, even better swimming, hunting, and predator avoidance abilities as they grow larger. And the other thing that really impacted fish during the heat wave, and will continue to be a huge challenge moving forward with climate change, is the fact that, being cold-blooded, obviously the aerobic scope, or the potential for growth, the metabolic rate, they all vary with temperature. So they will increase to a point, a fish can have a higher metabolic rate, grow faster if there's enough food, perform well to some optimal point, after which, even with more food, it's not going to go well. And then there's just a decline in the ability to maintain their physiological processes, and they can't handle it. Although this can shift. There's some variation in populations, and there can be selection for warmer optimum or lower, depending. And so that's where you can imagine selective mortality can take out fish that aren't handling it as well, and those that do survive to the next generation might have a slightly warmer, say, T optimal temperature. So all of those are relevant to the Pacific Cod in the Gulf of Alaska, $103 million fishery, one of the most important fisheries in the region. But given that 2014, 15, and 16 heat wave event, the cod fishery completely closed. So there's a 71% decline in the population by 2017, and it was a historic, unprecedented shutdown, complete shutdown of the fishery, which sadly, as often is the case, the more threatened or in crisis something is, then we actually have more resources to study it and understand it. So that led to a lot of research still going on to understand what happened, and can we do anything about it? How can we predict what's going to happen next? And sort of the three primary factors. One was a decline in spawners, in the sense that this graph is female spawning biomass. So the number of females from different NOAA surveys in the Gulf of Alaska from 1977 to 2019. And you can see how at the end of the heat wave each year is getting lower and lower and lower until it was what they perceive, that they conclude is a threshold level where it's just not beyond, it's below optimal for any successful reproduction. There was some reproduction, but what happened is they were larger, but many fewer juveniles in the nursery grounds. And this Pacific cod settle in coastal nurseries. And here you can see from 2007 through 2019, this is July, fish size, little beach stain surveys off Kodiak Island. And then those fish after the marine heat waves were bigger, but there were way fewer of them. And the graph on the right here shows you number of Pacific cod per beach stain set. With this great recruitment year, a strong cohort in 2012 with over 150%, and then barely any at all during the heat wave, and then a resurgence in 2017 and 18 in terms of numbers, but they're still big. So something's still going on there. So Pacific Cod have a pretty interesting and complex life cycle that the parents, the adults, go down to depth around 150, 250 meters, where they lay adhesive eggs on the bottom, and they are usually found around 45 degrees Celsius. And then when they hatch out, they pretty quickly go to the surface, where they usually interact with waters that are around 6 to 8 degrees when it's not a heat wave. And they feed on diatoms and then copepods as they get larger. And then they will settle in June, early July, into these coastal nursery habitats where normally temperatures are around 10 to 12. And then those that make it go on to reproduce and be part of the fishery. But what's really interesting about this species, and it's referred to as a stenothermal, a very narrow thermal tolerance. And so lab studies have shown this is temperature from -1 to 16. The proportion of eggs or embryos that can successfully hatch is less than 20%, except in this little window from 45 degrees, really narrow. So that's clearly a bottleneck for this species. And so it's likely that there was a reduced hatching success during the heat wave, one sort of the potentially reduced female spawners, and then reduced hatching success for the ones that did spawn. And then we have been working with the North Pacific Research Board to then look at larval survival and growth and juvenile settlement and foraging for those that did hatch pre and post heat wave, to better understand how the heat wave affected their hatch. The timing, a big question were those big juveniles that were arriving in the nurseries, were they hatched way earlier? Did they just grow so much faster because of the heat wave? Was it a combination of both? And so this builds off of beach sand collections on Kodiak Island. I should have a little inset. Apologies for not having that. This is sort of the north portion of the Gulf of Alaska, and this is Kodiak Island. And this is where the beach sand sites are here, and the larval sampling done by ship is in that blue grid. And we have data from 2006 or 7 all the way through 2019 of both the larval and the juvenile, so the different life stages. So as I mentioned, there were those larger but many fewer juveniles in the nursery. And so one of the primary things we're sleuthing out here is, are they older? Were they growing faster? Did selective mortality just lead to the largest fish, or some combination? And so I've been talking about heat wave designations and heat wave years, but I'm going to point out here is the blue box is 2006 to early 2014 through 2014, and then 2015 and beyond. And even though technically 2017 and 18 were not heat waves, they were, as you can see, warmer. They were positive anomalies. And what we see the fish doing is that they still clearly are responding to the warmer waters. So I'm mostly going to talk about pre and post heat wave from here on out. And so how do we do this? How do you figure out when a little 3 or 4 month old fish actually hatched? You might be familiar with this if people have worked on salmon or collected otoliths for the state or for other projects. We do a lot with otoliths, otolith chemistry, otolith structure, their balance and orientation organs. Here's a picture of them attached to the nerve that communicates with the brain, the left and the right. But scientists love them because they grow a little bit every day. And so if we take that otolith and polish it, in this case, polish out the middle, we can get a chronology, a growth chronology of every day of the life of that fish, and the width between those daily increments is related to its body growth. So we can create a lifetime growth trajectory at the individual level, and we can determine the age of the individual and the day it hatched. So we really can go back forensically and say, okay, for those fish that survived, what did you do? How did you do it? And so what did we see? Here, the pre-heat wave in blue on the left. Remember, so the fish were smaller and about 7 millimeters smaller than post-heat wave. And we determined that they did indeed hatch much later before the heat waves. So a mean hatch date of about mid-March versus the 7th of April. So 21 days there. with the biggest difference between years, April 16th to March 1st, being almost 40, 45 or 46 days. So really dramatic timing. So the larvae were definitely hitting the surface waters in very different times due to a variety of factors related to the heat wave. So on average, they were 3 weeks earlier since the heat wave, even including quote unquote non-heat wave years. And that maximum difference was 46 days. So it's 6 weeks, which is pretty dramatic shift in just a year or two in terms of what's going on in the phenology of the fish, the timing of the seasonal reproductive events. And so we're curious on disentangling what's driving that shift. And so one of the things we looked at was how temperature during incubation, right? They're supposed to be at 4 to 5 degrees, but we do have some bottom temperature data we can use to estimate what the temperatures they actually experienced are. And so this is the mean incubation temperature at 100 to 250 meter average in the Gulf of Alaska. And the number of days it would take at those temperatures to hatch. So you see 2007, one of the coldest years, earliest years, is almost a month. But then there during the heat wave, you're just at 16 days, almost cut in half. So obviously the direct effect of temperature can account for about 25% of that shift that we saw in the hatch dates. So that's part of the story, is that even though there was some 4 to 5 degree water, it was closer to 5-ish instead of 4-ish. Cumulatively, that was accounting for about 25% of the shift just because they were developing faster.
Can I ask a question about that?
Yeah, please do.
Okay, yeah. So just the warmer temperature increased their embryonic development rates, is what you're saying. They just, because the water temperature was warmer, they were able to develop to the point of hatching 6 weeks faster?
Well,
were they laid earlier too? Do we know?
So perfect. So what we found is that of that 21 to 46 day difference, this effect of having warmer temperatures during incubation could only account for about 25% of that shift. So it's part of it, and it's important, but it's not all of it. Exactly. So it appears that that can speed them up a little bit, so it can account for some of that, which exactly asks your question is, what else is doing it? Likely it could be a combination of selective mortality, where say in this case, the later hatchers weren't surviving as well, or the earlier hatchers during the heat wave did survive better. So you had some differential mortality, selective mortality, that led to those hatch date distributions for the survivors. And or the parents could have moved their shifting. They shifted their spawn timing from, that mid-March into late January and early February. So it's likely a combination of all three factors, that they had a more rapid incubation development. There was some selective mortality due to the really stressful conditions and the high metabolic demand from the warm waters. And the parents might well have not only had fewer females, fewer spawners, but shifted their timing in response to the warming. So there's a bit of a coherent correlated response, which is good, right? If we're going to have any adaptation to climate change, that is a good part to the story. Yeah, great. Thanks for the question. Yeah, anybody else jump in? It's one of the other things, and again, I think obviously the heat waves were very acute, right? They came on fast, things had to respond. Obviously Pacific Cod did not fare very well. But you would think, even though the accelerated rate of change that we see in our ecosystems due to climate change, there's still, it's not going to be that fast, right? There will be a little more time to adapt. And this is an example of the match mismatch hypothesis. So what we're able to do is, for the larvae that were collected in May, we looked at their hatch date in relation to a metric of the initiation of the bloom. So when does the chlorophyll, the phytoplankton, when does it start to grow and expand in the Gulf of Alaska? Well, when it happens later, like at around day 120, it was well correlated with the hatch date. And we see it very, very well correlated. In the juveniles down here on the bottom, we see a similar positive relationship. It's not as strong, but a lot's happened. That's another 2.5 months of life and death and moving around. So this is an example of how the temperature response, right, is the algae and their their production cycles are correlated with temperature, as is the hatch timing. So there is some adaptive mechanism for the system to covary to the point where some fish still survive and potentially can contribute. And so the last thing we can do then, or that we're doing right now, is looking at that growth trajectory then. So we know they hatched earlier during the heat waves. They were into warmer waters. We're exploring, other than phytoplankton, what kind of zooplankton were in the water and how that influenced it. But hopefully what you can see here, to me, the very cool thing about being able to get these individual growth chronologies is you can then ask questions where you're separating what we call ontogeny or age from day of year. So we can align all these growth chronologies. Each one of these is more like 80 fish from multiple years during the heat wave in 2017, and 18 is the interheat wave or the preheat wave, the years before 2014. Or we can align them by day of year. And so here, obviously, we're comparing all fish that were 1 day old with all other fish that were 1 day old, and here all fish that were 30 days old. And what we see is that the heat wave had negative effects on early growth, but then for those that survived to a month and beyond, they were growing faster during those heat wave years. And similarly, then we can also just look at how in the bottom graph we can understand how conditions in May, say, or the timing of the bloom was related to the growth separate of the age of the fish. And so that's helped us understand sort of the relative contribution of that shift in hatch phenology and that increase in growth rate. So to sort of sum it all up, right, in stressful conditions such as a heat wave, which is really stressful for a Pacific cod, which is a pretty stenothermal fish, it likes its narrow window of temperature. Mortality is higher for those early life history stages, and there's that 4 to 5 degree window. So there's mortality of those lower egg success. And then the larvae, again, going to have probably higher mortality, increased metabolic demand, some selective mortality, leading to those larger juveniles, but lower densities. Only the biggest, the fastest growing, sort of the biggest and the best survive. And what's interesting about the Pacific cod story is up through 17 and 18 even, we weren't seeing a shift back to later hatch dates or smaller bodied fish. And just collections in 2021, just last year, showed a really interesting distribution where we got both cohorts. So in the size distribution, there's the early and the late, all in July. We're having little fish, like smaller than before the heat wave, and bigger fish, bigger than since the heat wave, which is a really interesting phenomenon. We'll be aging those and trying to understand that, because that suggests that that could be adaptation. That could be the population responding. Some individuals are shifting. Some individuals are staying more in the old manner. So it's more to come on that story. I want to shift on a little bit to salmon. So maybe I'll just stop and see if anybody else has any questions or thoughts about Alaska or Pacific cod before I kind of come back down into the California current.
I mean, I definitely have a couple more questions.
Sure.
So is it possible that there's also a shifting in spawning time based on temperature as well?
Yeah.
Like what's like the cue for initiating spawning?
Yes. Sorry, that was the
Oh,
sense of
yeah.
this graphic. Yes. So, and that's another thing we're just now been talking to the Alaska Department of Fish and Game, because it's a little tricky. They fish on the spawning populations. That's often when they're harvesting in winter. And so we've tried to look at whether you can get anything out of the catch data or the timing of a peak harvest. But what we're shifting to now is the state, ADF&G goes to the plants and they sample the totes that come in from the different fishing boats. And they've got a long-term archive of sex, and they stage the maturity of the individuals, the females. So we're going to work with them to go back in that time series and see if Are we seeing a similar shift in when the adults are spent or have finished reproducing as we see in the larvae? Is there any correlation there? Is that part of the shift? So I think we should be able to quantify that with their help in the next few months. But that's probably part of the story, but no one really knows how much of it, how much they're responding that way. And what's interesting too about the cod is there's evidence that a lot moved north, like all the way into the Bering Sea. So there were more Pacific cod caught in the Bering Sea in 2016 than like ever in history. And so there's a big question of, are they going to come back? What's going on there? Was that some weird shift in where they spawned, not when they spawned, but maybe where they spawned? So there's still a lot of missing pieces to the cod story. But yeah, good question.
The other question that comes to mind is, are you looking, or is anybody in your lab looking at DNA diversity and whether or not this is operating as a bottleneck event, as far as like fish that were adapted to that were already in the population spawning a little bit earlier were the ones that survived, but now it's not returning because that's the population that survived the blob?
No, it's a great question. And there are, so I think the lab doing the most work right now is NOAA's Alaska Fisheries Genetics Lab.
Okay.
And so they have a marker set that looks at sort of the neutral genetic structure, of which there's more than people have thought throughout the Gulf of Alaska. So you could have different stock, different paternal stocks contributing with slightly different phenologies. There's also a gene called Zonapalooza 3, is EP3, which has recently been shown to have some pretty dramatic spatial distribution in the Gulf of Alaska, where one haplotype is found only southeast and the other one is found northwest. And so the idea is that could have something to do with successful fertilization and related to the thermal environment. So there are efforts now to try to link up that ZP3 gene with hatch success and larval performance. So yes, I think hopefully relatively soon there'll be a little more knowledge of what's going on there.
That's really interesting. Thank you.
Yeah, great questions. Well, I'll finish up a little bit with salmon. So this, I don't know if anybody's seen this. If you have, put a little like thumb up in your picture, but this is from NOAA and it's called the stoplight chart. So this has been around since, I think the stoplight chart was developed in about 2005 or 6. So a Northwest Pacific Fisheries Science Center and OSU colleagues, they do a surveys funded by the Bonneville Power Administration since '98 to understand how ocean conditions are influencing, in this case, mostly ESA listed Columbia River stocks, salmon groups, mostly focused on the interior spring Chinook, which are the most threatened. And so that survey happens, it used to happen in last week of May, June, and September. And then it got shifted to May and June. And so that goes out and builds off the NH line I mentioned before. So there will be monthly data collected there, and then these synoptic surveys that go all the way up to northern Washington, La Push, where nets are trawled, juvenile salmon are collected, bongo nets, we collect the zooplankton, there's phytoplankton, and they come back to the lab, and then they're big fish parties. 20 or 30 people will cut, a piece of the salmon for genetics. Some will take the stomach to look at what they've been eating. I look at their otoliths. Sometimes we look at stable isotopes or markers in their tissue that tell us things about how they've been eating. And so this time series has been going on since '98, as I mentioned. And this has turned into sort of the management tool that synthesizes the science that's observed in the ocean. And so just like a stoplight, red stocked salmon bad, yellow, uncertain conditions, and green, all light, all by all expectation, those are good conditions for salmon. And that designation is based on a variety of studies that have been correlated with survival to adult, small to adult returns. So there's a suite of climate and atmospheric markers, the Pacific Decadal Oscillation in winter and spring, and the ONI index. sea surface temperature at different buoys offshore, upper 20 meters, temperature in winter and summer, deep temperature, deep salinity. I mentioned the copepods earlier, so this is here the same index I showed you, the copepod richness or the number of species, the biomass anomaly of the northerns, the biomass anomaly of the southerns, which is bad. The biological transition is when we switch from those downwelling winds and then all of a sudden it's upwelling and the north wind is blowing in your face, that it actually happens at quite a predictable time. So, and a little earlier is always good for salmon. So the timing of the biological transition. The nearshore ichthyoplankton is an estimate of the forage fish I mentioned, the anchovy and the sardines that they like to eat. And then actually estimates of how many were caught. Again, the critical period here applies to early ocean residence is considered a critical period in salmon. And so when it's a bad year, not many are caught offshore. When it's a good year, often we catch a lot. So that's actually a pretty good indicator. And then all those metrics, all those indicators are ranked. And here you see the mean of the ranks and the rank of the means. And you can picture a bunch of scientists and statisticians arguing about those, which is why they're all there, in addition to a whole nother suite and different ways of analyzing this. But what you can see here is that there's the beginning of the blob. 2015 and 16 is when it hit our coast. And that was the 22nd and the 24th worst year for salmon. 1998, if anyone was around that long ago, was the really bad El Nino, which was the 23rd worst. So bad. The blob and the marine heat waves are not good for salmon, which is probably no surprise. And a lot of times the marine heat waves are correlated with drought conditions in the Pacific Northwest, and climate models predict that those will occur with increasing frequency as well. There's some good news here, right? Because if you're looking at 2021, which was last year's outmigration, a lot of green here. And it's the second best year in terms of indicator after 08. So, you know, they swim up to Gulf of Alaska, things can happen there. But in general, the predictive capacity of this stoplight chart is not bad. It's as good or better than the sibling regressions. It usually adds some understanding. So at least in the short term, it's looking pretty good for salmon, and we will see what's happening in 2022 this month, in fact. So I'm just going to end by, it's probably obvious a little bit, hopefully, that there's some winners and losers when it comes to things like marine heat waves. And there will be, probably with most aspects of climate change and variability. And so sadly, some of the winners include the toxic phytoplankton, which obviously is not ideal for the human system or a lot of the coastal mammals. And the tropical, subtropical copepods, market squid actually did really well. Rockfish, for reasons I don't think are entirely clear, increased recruitment during the heat wave events. And it was great for tuna fishing. I know people that tuna fished in the Gulf of Alaska. So there you go. Sometimes things can turn around. And orcas seem to do pretty well. Whereas, as we talked about, the groundfish, salmon, Dungeness crab, the subarctic cold fat copepods do not do well. And so I think it just highlights the fact that as we move forward, we'll be seeing more of these trade-offs and winners and losers as the ocean responds to climate variability. So I'd be happy to take any questions.
I have a question.
Yeah.
Back on that stop and go chart you showed, the period from around 2005 to 2006 or 7, was there an explanation for the rise in those temperatures, or for the red stoplights?
Sorry, I shouldn't have stopped sharing. I was too quick. 2000. So say again, 2007. Let me pull it back up. I'm sure I can figure this out.
I'm looking at it now.
Oh, I still am sharing. Good. Okay. Excellent. Let me.
Yeah, the section where it looks like it's. You have to bear with me because my eyes aren't the best.
Yeah, I'll make it bigger there. Oh gosh. Okay, close your eyes, everybody. Don't puke. Back down here.
Here she comes.
There we go. Okay.
Yeah, 2000. I guess it's 2003 to 2005 or 6. There's a large cluster of red
Yes.
stoplights there. Was there an explanation for that time frame?
Yeah, it was a positive PDO. So the Pacific Decadal Oscillation is an atmospheric pattern that, well, it used to oscillate more regularly, but again, it's negative and positive anomalies. And in the Oregon and Washington waters, when it's a positive PDO, we are warmer than normal, and Alaska is actually cooler. And then it reverses in the negative PDO. So negative PDO for us is good. Positive PDO is bad. And there was a multiple years of positive PDO followed by what people thought was an El Nino in 2005, but it actually was a sort of again an anomalous warming event, where there was an El Nino in the Central Pacific, but the manifestation, which is a big Kelvin wave which travels up the coast and brings us warm water, never got to our waters, but we were still really warm. So a combination of positive PDO cycle and some other what they call ocean atmospheric teleconnections that increase the warming off our coast, but not in the direct El Nino way. So yes, those were similar to like, so in 1977 was the big shift where we switched the PDO switched signs. And that was the big prediction, like we can figure out salmon, all we need is the PDO. Positive PDO and negative PDO, and we can predict it all, which was kind of true for a while. But the flipping of the cycles increased to be 3 to 5 year. Now they're called semi-decadal instead of Pacific Decadal Oscillation. And that signal seems to be weakening. And most model predictions suggest that it's this interaction with climate change is just changing the way the ocean is responding to that atmospheric forcing.
Thank you, Dr. Miller.
We've got a question in the chat here. The question is, have spring Chinook arrivals at Bonneville exceeded expectations this year? And I'm not tracking those this year, but are you?
I have not yet. I've been thinking about Pacific Cod a lot for the last few months. Let's see, it's 2022, so 19, 20, 21, 22. I wouldn't, well, well, we'll have to see. I do not know. And so the spring Chinook leave as, you might know this actually, right? They leave as yearlings. And so they come back, most of them come back 2 years afterwards. And so the 2022 returns went out in 2020, which was not a great year, but they have surprised us for sure. I mean, the one interesting thing about all these recruitment studies is it's pretty easy to predict a really bad year, and it's pretty easy to predict a really, really good year, but like everything in the middle is often a lot harder. And usually there's like one mechanism maybe they don't quite figure out. So I will look tonight and email Zach, let you know. Well, thank you. I hope a trip to Alaska was an okay thing for the Nehalem Watershed Council. Appreciate you coming out on a whatever day it is, a Thursday night.
Thursday night, I think it is, yeah. Yeah, I enjoyed this tremendously. I did spend a little bit of time in the working for as a ground fish observer for a bit off Oregon, Washington, and so I've caught a lot of Pacific Cod, and so this was really interesting to kind of look at, the numbers that you're presenting and line it up with the dates that I was doing that work and thinking, oh, okay, that's how it all fits in, So that was kind of fun.
Excellent. Great. Well, thanks.
Yeah, thank you so much. This has been great. If we don't have any other questions, we have lots of thank yous coming in the chat. But if we don't have any other questions, I think we can call it a night for tonight. Thank you so much for being here. And this actually is the end of the Watershed Council's speaker series for this season. And we'll be back in October, and there'll be announcements about our speakers and who we're going to be having closer to that date. So thank you everybody for being here, and we'll see you in October.
Thanks, Zach. Nice to meet you.
Thank you too. I appreciate it, Jessica.
Bye-bye.
Bye-bye.